Good afternoon. My name is Stephan Holl. I'm the CEO of Conscio Group, and I will be your host this afternoon. I will only share a few opening remarks, but we are really excited to start our webinar on from analytical similarity to clinical data, tailoring evidence for biosimilar approval. It's great to see such immense interest in today's topic. I think it's just a clear testament of how timely and important the discussion is. And, you know, we do this during a time in which we witness probably one of the most significant regulatory changes in biosimilar development over the last decades. So that's very exciting, and we are very happy to engage in a lively discussion with you today. So it's been about 2 months now, on April 1st, that the EMA, the European Medicines Agency, published this draft reflection paper that has really, created quite a lot of discussion in our industry. So it's really just not another guidance update, but it represents a fundamental paradigm shift in how we approach biosimilar development and approval. And it marks also the first time that the EMA formally acknowledges that comparative efficacy studies may no longer be mandatory for biosimilar approval if and when sufficient analytical and pharmacokinetic evidence demonstrates biosimilarity. The reflection paper also embodies a principle many of you have advocated for quite a long time, that it's the structure of biosimilars that determine the function, and that when we can rigorously demonstrate structural and functional similarity through advanced analytical techniques combined with robust pharmacokinetic data, we may have all the evidence needed to ensure patient safety and therapeutic equivalence. So those implications are pretty profound. Current estimates suggest that the efficacy studies account for almost half of biosimilar development costs, with phase 3 efficacy studies potentially adding also another 18 to 24 months and tens of millions of euros to typical development programs. And by potentially eliminating those, we are looking at a future where development timelines could be reduced, the barriers to entry can be lowered, and ultimately patient access to life-saving biological drugs can be significantly improved. One example that I found particularly staggering was also published by IQVIA a few months ago, which said that about 120 biologics are expected to lose patent protection over the next 10 years, and yet 90% of those currently have no biosimilar in development. And we believe that, with this new regulation, this could be significantly changing, and we could help in partnership with our customers to increase access to some of those treatments. So, what makes this shift possible is a pretty remarkable advancement in the analytical technologies we've been using. They offer unprecedented precision in characterizing biosimilars and reduce, in general, our reliance on clinical efficacy studies that, in history, and that has also been demonstrated. So some of the research of the EMA, they have just failed to provide meaningful additional information beyond what was already proven through the analytical characterization of biosimilars. And the EMA now officially supports this evolution, as the efficacy studies results have simply confirmed what comprehensive analytical and pharmacokinetic assessments had already previously established. That was one big part of the discussion in our last webinar. So today we are exciting to have assembled, a true expert panel who will guide us through the practical implications of this regulatory evolution, as we are now, in a phase where still feedback is being provided, and we'll try to shed some light into the dark and really encourage a discussion with some of the most leading experts in the field. So we explore how to leverage extensive analytical similarity for regulatory success. We examine strategies for building robust scientific packages that can support this new paradigm. And we also discuss how to maintain the high standards of patient safety while embracing these streamlined approaches for clinical development. Our speakers have decades of combined experience across those different topics, and they'll share their insights in how to navigate this transition, identifying when the tailored approach is appropriate, and building compelling scientific narratives that regulators can confidently support, because we believe that will be one of the key things to get right now and to get our heads around, how to basically agree with regulatory authorities on clinical development paths. Just as a side remark, so the consultation period for the reflection paper extends until September 30th. We hope, that we can stimulate some of the thought process around giving feedback to the email also today. And we encourage you to put your questions today in the Q&A section of our chat, not the chat function, the Q&A function of Teams, where we can then selectively bring up some of those questions at the end of each speaker's slot. So therefore, we definitely encourage you to participate in our discussion. We'll thank you for the attendance. And now, without further ado, I'm very happy to hand over to our first speaker on the analytical similarity, the future of biosimilars, leveraging extensive analytical similarity for regulatory success. Thakara, Rama, Velisetty of IQVIA. Thank you for joining us today.
Hi, everyone. My name is Dr. Thakara. I'm working in IQVIA as a biologics area lead. And at the CMC Center of Excellence, we support different clients across the globe for their development of biosimilars. At IQVIA, we have end-to-end solutions that we support the clients right from the pre-IND meetings and then clinical trial phase 1, 2, 3. And finally, we also support them for marketing authorization for different regulatory agencies. So today we are going to discuss some basics and then fundamentals. And as Stephan told, the EMA reflection ideology and how they are thinking, what is the EMA thought process on leveraging the particular extensive analytical similarity for the approval of biosimilars. So today's content would be on the analytical similarity. We'll touch base with the definitions and the overview for the people who are new for this area, and then importance of this analytical similarity and why it is crucial in the biosimilar development after knowing the different agencies' thought process on that, and then analytical techniques, which are very key now for demonstrating the structure-function relationship between the proposed biosimilar and the reference product. So we will also discuss about the overview of state-of-art analytical methods which are used in this analytical similarity. And we will also summarize the EMA recent reflection paper insights and how it is linked to analytical similarity assessment. And we also discuss very few case studies and example in which there is a chance of getting phase 3 waiver, how to attain that phase 3 waiver, followed by question answers, and we will be touched base with. And if you see the definitions, of biosimilars. I hope everybody knows it, but since it needs to be touch base, the basics, so we are explaining. So the FDA definition is almost similar to EMA definition, but only the difference is that a biosimilar is a biological products which are prepared, which are manufactured using live cells, and that product is highly similar and to the particular FDA approved reference product. And if any differences observed between the proposed biosimilar and the FDA approved reference product, those differences you need to prove that they are not clinically meaningful. So this means the proposed biosimilar must match with the reference products in terms of safety, purity, and potency. Purity and potency in other way, quality. So in terms of safety and quality, if proposed biosimilar is similar to your reference product, then it is called biosimilar in terms of FDA. Whereas EMA, the same concept they have put together, but they are referenced from that European approved product, which we call as RMP, reference medicine product, and the proposed biosimilar should be equal or similar to your European reference medicine in terms of structure, in terms of biological activity, efficacy, safety, and immunogenicity profile. So if you see the right hand side, we have given some similarities of the reference biologics and biosimilarity. So these biosimilars are, the concept is different from generics before. Now the agency's thought process are coming towards the biologic generics. So which means we are getting waivers when you prove structure function relationship of our proposed biosimilar to reference product. So when we check the reference biologics and biosimilars, both are complex in nature. They are protein in nature. They are glycans involved, and there is so much complexity involved in terms of structure. And then it has so many functions to prove. So both are complex, and it must be proven that the primary structure of both the reference biologics and biosimilar candidate should have the same primary structure. And anyway, as we will provide a Q2PP, we will define a Q2PP quality target product profile in which we propose same strength and dose just like RMP. And as part of Q2PP, the same route of administration we will propose, and with the same mechanism of action, it needs to be proved with both the molecules. So the reference biologic product is already proven efficacy and safety, hence it is in the market by the approval. And the biosimilar, we prove the similarity to the reference biologics in terms of structure and function, and also safety studies. Coming to the concept of analytical similarity, and very hot topic nowadays because of EMA reflection. So what is analytical similarity? So the analytical similarity, it refers to the comparison between the proposed biosimilar and reference medicinal product in terms of by employing a range of analytical techniques, especially state-of-art analytical techniques. If there are any quality attributes, if needs to be confirmed that there are some differences, then orthogonal verification with another analytical methods which are employing different principles than the previous analytical method also to be employed. And so when you employ a range or battery of tests for both the products, side-by-side analysis, and when you do this activity, you must ensure that the biosimilar is highly similar to the reference product in terms of structure and function relationship. In terms of structure and function relationship, if you see the right-hand side, there are two monoclonal antibodies. One is reference medicine product, and another one is proposed biosimilar, wherein which you need to prove the similarity between these two in terms of so many physicochemical properties and biological properties. So when you prove the similarity in between these two molecules, so the agency thinks that when they are structurally similar, when they are functionally similar, it is not necessary to conduct clinical trials. So that is their approach and their thought process now, which is very welcome, and it is going to change the world very soon. So before going into the analytical similarity assessment, so let us discuss about how this analytical profile to be defined. So initially, we select quality attributes of your product. So quality attributes are nothing but the property of a product. So the physicochemical property or biological property or immunological property, even microbiological property. So these properties we call it as quality attributes. So these quality attributes of any product, we will perform a criticality risk ranking assessment by applying ICHQ 9 guidelines. When you applying each property of a product, be it proposed biosimilar in our case. So the properties, when you assess criticality ranking by applying Q9 principles like PHA, preliminary health, hazard analysis, or there are different tools available in ICHQ9. So basically, for biological products, we apply the criticality to 4 pillars, which we call it as safety, efficacy, and the PK/PD and immunogenicity. So we perform risk assessment on these 4 pillars, and then based on the outcome of this criticality risk assessment, we classify them as into critical quality attributes or a quality attribute or non-quality attributes, non-critical quality attributes. So all critical quality attributes must be thoroughly characterized by using a series of multiple analytical methods. So once you assess the quality attributes of your product, then you will perform analytical similarity evaluation by creating a protocol and proposing a acceptance criteria in how both the molecules are compared and how the conclusions to be made. All will be defined in the protocol. And after execution of the protocol, you will be summarizing the results. During the summarizing your results, you need to apply some statistical analysis. So this is very crucial nowadays, like how you are portraying the comparison between these two proposed biosimilar versus reference medicine product. So the FDA initially had 3 different tiers for the comparison. Like they have tier 1, tier 2, tier 3, like the visual comparison, and then quality range, and then equivalence testing. So these kind of approaches they proposed. Now they withdrawn the proposal of these 3 kind of statistical tools that they proposed initially in their guidelines. Now any sound statistical analysis that is proven, and when you have a strong justification, you can propose your statistical tool and approach which you employ for your analytical similarity assessment. So nowadays, majority of companies, as per my experience, they are using equivalence testing and then tolerance intervals and plus or minus 3SD approach also to compare both the molecules, both the products. So if you see the right-hand side, it is just an overview of your analytical package, and we will discuss anyway the analytical methods that are used in similarity assessment and comparability assessment. So if we have different properties for product, like physicochemical properties, biological properties, and immunological properties. So preferably, we will discuss each one in the next slides. This is the critical quality attribute slide. wherein which we thoroughly need to assess as per ICHQ 6B guidelines for the characterization of both the molecules and also the comparison in terms of analytical similarity. So primary structure, like the amino acid sequence confirmation, intact mass, and then subunit mass, and glycosylated mass and a non-glycosylated mass, and then peptide mapping, fingerprinting, or peptide mapping for identity. So primary structure and higher order structure and post-translational modifications such as glycosylations and any other post-translational modification that we observe, oxidation, deamidation, such kind of PTM as per the molecule that we are developing, and product-related impurities and process-related impurities also sometimes they might be asking nowadays, but does not necessarily to compare these process-related variants in the analytical similarity assessment. However, the main key biological property, physicochemical properties, biological properties, and immunological properties, we need to focus and then compare between these two molecules. So when you see the higher order structure, you have secondary confirmation, secondary structure confirmation, tertiary structure confirmation, and then other confirmation stabilities like stability indicating parameters of the particular molecule. All that properties needs to be characterized. And then when it comes to glycosylations, since we all know that the monoclonal antibodies are glycoproteins in which there are glycans also part of it. So these glycans have multiple functions of doing FC related functions, and then it can also give a type of more serum half-life or plasma half-life. There are so many different uses of the glycosylation. And when you are not controlling this glycosylation during the production, and if the glycosylation are not similar to reference medicinal product, then agency might be asking so many questions and so many additional studies you might be requiring. So it is very essential for the developers that this post-translational modification glycosylations to be in control while manufacturing their product. So the glycosylations we generally perform with oligosaccharide pattern, and then there are different kinds of monosaccharides which are forming these glycosylations. So we need to identify different monosaccharides which are present in the glycosylation, and the pattern of the glycosylations also we need to identify and prove percentage of each glycosylation forms. So coming to product related variants, wherein which we call them as a variant because they are not impurities, but they can show the difference between our reference product and the proposed biosimilar when there is differences happening in the manufacturing process. So here the charge variants which everybody knows that the each monoclonal antibody is made up of different amino acids, and amino acids do contain the charge. Based upon the composition of the amino acids, which is outlaying the particular molecule, the charge would be changed for some monoclonal antibodies, which are manufactured in the same batch. So the charge variants also to be in line with the reference molecules. So here are basic variants, acidic variants, and neutral variants, which call them as a main variant. So all these three variants also should be in line with the ranges of a reference medicinal product. That is what the agency expects. And then coming to aggregates and fragments. So these are generally called as impurities. We do not expect to see them, and your process should be able to clear them before it reaches to the final DP. So the aggregates wherein which two different molecules, two monoclonal antibodies or multiple monoclonal antibodies clubbed together and they form high molecular weight impurities. And the fragments, that the example I'm taking monoclonal antibody, so sometimes the FAB portion, FC portion can be cleaved, and heavy chain and light chain can be separated. And non-glycosylated heavy chain also gets separated sometimes during handling, mishandling, and then during even manufacturing process itself, it can happen. So all these type of fragments and aggregates, which we call it as the main impurities, that should be cleared by your process. And the final end product impurity should be similar to your reference medicinal product. That is what agency expectations. So when you see the analytical package, so you need to employ a state-of-art analytical methods, right from HPLC, LCMS, which there are different LCMS again, ESI on top, MALDI-TOF MS. So there are different LCMSs where we determine the structure of protein and also determine the glycosylation pattern. And peptide mapping, we employ different instruments like HPLC and again MALDI-TOF and LCMS and MS. And when it comes to secondary structure, we use FAR UVCD and then FTIR. And when it comes to tertiary conformation structure, we use near UVCD, and we also employ FLR, NMR, and again XRC. All these high-end instruments are very sensitive in determining the differences between two molecules and even to characterize the particular product. When it comes to conformation stability, there are multiple options, but we use DSC as a standard method. And you can also use NMR for the conformation stability, where the destabilization property at which temperature we will attain. So TM1, TM2 we used to call melting temperature 1, melting temperature 2, at which temperature the product is able to destabilize by its own. So that is also agency expectation. And when it comes to glycosylation, so this glycosylation usually we do with RP-HPLC with the fluorescence detector and Hillick HPLC, and we use LCMS MALDI-TOF also. And some companies use normal chromatography also. Based on the identification of these standards, we'll compare the standard peak with the molecule peak, and we compare both and provide the conclusion. And when it comes to aggregates and fragments, there are multiple methods. SEC, which we call size exclusion chromatography, so, SDS page, which is preliminary test, and then you have SECMALS, and then you have EM, AUC, ultracentrifugation. All these different techniques can be employed to determine percentage of aggregates and fragments. And when it comes to charge variance, it is cation exchange chromatography, anion exchange chromatography, isoelectric focusing, electrophoresis, and then CZE. And we also use QTOF MS sometimes to determine charge variance. And related proteins like these are called the proteins which are coming from your process, or the proteins which are coming out from the purification resins. So this will be also, we will be checking using some different chromatographies like affinity chromatography, boronate affinity chromatography, and in case of a protein A, we will do ELISA technique to determine protein A, which is present in your product, which is coming from your purification process. So these are all called state-of-art analytical methods, which are able to determine very sensitive differences, very small, minute differences between two molecules. That is the reason that EMA is focusing on employing this analytical methods to prove the structure and function of both the molecules are similar. So, and here I am with a case study on analytical similarity, and this is one company that has published their data on public platform. So if you see here, I'm just focusing on two quality attributes, the primary and higher order structure. So where they have performed molar absorptivity, peptide mapping, intact mass, free thiol analysis, and DSC, as I informed TM1, TM2, TM3. So these are the different analytical tests they employed to prove primary structure of product. And these are the quality attributes that they have taken, like molar absorptivity, extinction coefficient, deamidation, oxidation, and some amino acid modifications like pyroglutamate modifications, N-terminal and C-terminal lysine truncations. And they also determined intact mass of glycosylations separately. So all these different analytical package or battery of tests are performed to prove primary and higher order structure. And then they concluded the summary of their analytical process. If you see here, they used statistical tools like minimum and maximum approach and mean plus minus 3SD approach. Two approaches they have employed, and they have taken their own product, 15 lots of their product, and rituximab Europe region approved product 15 lots, and US rituximab 15 lots. So this is called two-way comparison. So the two-way comparison, they summarized all the results. When you check, all the ranges are falling between Europe reference range and US reference range of their product. So in that case, it is proven that the biosimilar structurally, example in the primary structure and higher order structure point of view, they are in line with the quality ranges of the reference medicinal products of both the Europe region and US region. So now let us focus on the EMA reflection paper, as we have seen different analytical methodologies that we use to quantify or summarize the complete analytical and biological properties of products. So now the EMA reflection paper proposes to the mankind that the robust analytical and functional comparability. If you have a robust analytical functional comparability, in many cases, it reduces or eliminates the need of extensive clinical efficacy trials, especially for biologics, with well understood mechanism of action. So this is the key. If a biological product is having a well understood mechanism of action, then structure function relationship, if you prove, that would be enough to approve the product. But if your product is having multiple mechanism of action which are unknown, then in that case they will ask for phase 3 clinical trials to be performed. So now, why this analytical similarity matters more now? As explained, the advancements in analytical technologies like mass spectrophotometry and cell-based bioassays and ligand binding, receptor binding assays we used to call. So all these critical things are SPR we use there, surface plasma resonance, BIA-core instrument we use. So all these advancements in analytical technologies allow for a high sensitive detection of any differences between biosimilars and reference products. So that is the key, that these instruments or analytical methodologies are able to find the differences if they are compared side by side by using the same analytical methods. And there is another reason that biological activity and clinical efficacy are increasingly inferred from structure and function relationship. So when there is a structure function relationship is proven, automatically the function of the product is going to reproduce its mechanism of action when it is introduced in vivo. So rather than requiring full scale of clinical trials, if you prove the functionality, and structure function relationship, that would be sufficient. That is the ideology of EMA. So now the streamlined development pathway, as I told in the initial, so you need to propose to the agency that the CQAs of your molecules are performed based on risk-based approach, and you have performed tier 1, tier 2, tier 3 attributes, comparison critical quality attributes, which are direct clinical relevance, and the attributes which less clinical impact on clinical relevance. Anyway, that would be categorized into non-CQAs or moderate CQAs. So this approach aims to reduce developmental costs. Automatically, the timelines also will be reducing. So this is streamlined development pathway that they are introducing. Now it will be beneficial for so many biosimilar manufacturers. And as I told, the statistical rigor still required. So different organizations employ different statistical tools to compare the similarity. But these statistical tool similarities should be prior to be discussed with the agencies that we are going to use these statistical tools. Would agency accept this approach? So if you have a clear communication between the agency and your organization, then automatically this particular analytical similarity is coming with flying colors. So that is very required nowadays. So we need to anyway follow statistical robustness and statistical analysis using equivalence testing, multivariant analysis, and then as I explained, there are different approaches. All approaches can be applied, and then you can submit your proposal to the agency. Yeah. Okay, so we discussed so many things about analytical similarities and then analytical methods, statistical tools, and then structure function relationships and all. To prove all this, how many lots are required? So this is a big question. So when you take broader RMP lots or more RMP lots, automatically the more will give more coverage to the variations. So until the reflection paper reveals, so the guidelines used to say the sufficient number of lots. So there was no clear number has given before, but now they come up with the number 15 to 30 lots of RMP are recommended in the reflection paper, which are adequately, which they think that it is adequate to compare. And proposed biosimilar minimum 6 lots they are expecting, but with my experience of 20 years, adding more to that 6 lots of proposed biosimilar will have advantage to cover more variation in the process and also in analytical instrumentation differences. So depending on the complexity of the molecule, these particular critical quality attributes, considering all these aspects, better to include more number of lots to compare with the more number of RMP. So usually I have shown here that this particular company or organization used 15 lots of all. So in that case, this particular number is statistically significant and it is adequate in terms of EMA and FDA. Hence, this product is got approved. So likewise, when a biosimilar manufacturer is trying to prove the analytical similarity, I suggest personally it should be more than 10 of your products, and then minimum 15 to 30 lots of RMP are required. And what the EMA reflection paper is lacking. So it is lacking something that of a specificity and statistical methodologies. So it is not giving any recommended statistical tools in that EMA reflection paper. That is the company's headache to prove that the statistical tool is sound enough to compare the two variations of molecule. So there is one limitation here, and an ambiguity around number of lots. Like they have given 15 to 30 lots of reference product, but it does not clearly define how to justify if you use a fewer lots in practice. So when there is less number of lots by any reason by the company than 15 to 30 lots, so what needs to be addressed, how it needs to be addressed, what aspects to be considered, that is not given by the EMA reflection paper. In another way, they are insisting to use minimum 15 lots by their statement. So geographic sourcing, they did not mention in the EMA reflection paper. By our previous experience, we should stick to the same requirement that if you are applying for Europe market, you need to outsource Europe approved reference products. And how to handle lot variability across different manufacturing sites. So the same Europe reference medicinal product can be prepared by different manufacturing sites of their own manufacturing sites. So if there is so much variation between RMP, then how to address that is not given in the previous guidelines and also in the EMA reflection paper. That is one limitation. And limited guidance on complex biologics. This is very important point that we should be noted. In the first slide, I mentioned that when there is known mechanism of action of any biological product, structure function relationship, if you prove, that would be enough to waive off phase 3. However, many recent novel biologics and complex biologics, when they are getting the particular patents expiry very soon, right from 2025 to 2035. In between, there are many biologics are getting patent expiry. So in those complex biologics, when there is complexity in terms of mechanism of action, this reflection paper is only applicable to well-characterized, less complex biologics, but it is not explaining about how to approach monoclonal antibodies, fission proteins, and other glycosylated products. So this is another limitation in the EMA reflection paper that anyway during this consultation in the public domain, they would be addressed. That is what I am thinking about EMA reflection paper. And case studies, I think everybody know about the waiver of phase 3 has been obtained for some products like insulins and GCSF, since they are small in size and well characterized and limited amino acid numbers. So they got phase 3 waiver. Example, pegfilgrastim, and then recombinant GCSF. And a notable example for monoclonal antibody is that adalimumab has got some benefits. Some submissions to EMA included reduced clinical data due to strong analytical package. So this is only the case I have seen, and in my experience, another product called nivolumab also got some reduced clinical data because of the excessive and extensive strong analytical packages that they submitted. However, full phase 3 waiver for MAB are still rare, and they are being evaluated by EMA case by case. This point to be noted. So the key takeaway of this analytical similarity concept is that EMA and FDA are increasingly open to waiving phase 3 trials, especially for well-characterized biologics, maps with straightforward mechanisms, which means if they have only single mechanism of action, whether with the FAB portion or FC portion, then this robust analytical data would be helpful in getting waiving of phase 3 trials. So this is the trend, especially for biological molecule, is a science-driven risk-based approach, that should be implemented right from the development of your biosimilar product. Thank you so much for your patience. And I'd be happy to answer your questions, if any.
In the Q&A section, very good insights into analytical similarity. You showed the data of 15 lots. What is the age of those RMP lots? Is it a head-on comparison?
Yeah, the age should be definitely to be considered when you're comparing this. So the particular age of reference medicinal product, you need to collect or you need to purchase the reference product in such a way that it covers its entire shelf life. Like, when it is freshly manufactured and when half of the half-life is done and the near to the half-life. So when you purchase 3 different ages of RMP, that would cover all the variations, and it is indeed necessary to purchase those RMP lots.
Then there is a question on whether it's needed to include clinical lots and commercial batches in the biosimilarity studies.
Exactly. So this is the point I missed. Thanks for reminding. So the particular biosimilar, proposed biosimilar lots. So what type of batches to be included in analytical similarity is that final commercialized intended scale manufactured batches, and preferably process validation batches. We call it as PPQ batches, and the clinical trial materials also to be included in analytical similarity assessment. And if you have any previous lots which are comparable to the commercially intended scale batches, then you can use them as a supportive batches also.
Right. It's a little bit the more the better, I suppose. The more robust the data set becomes as a whole. Okay, I think the next one is probably a bit similar. The lots mentioned 15 to 30 are analyzed as head-on analysis, or the data analyzed during life cycle of the molecule can be used for comparison.
I didn't get the question, Stephen.
Yeah, I don't get it fully either, but I think it refers a little bit to your earlier, the RMP lots. I think what you discussed, how we procure them and then analyze across the the different time points away from the production to make that comparison.
Yeah, it's about RMP. I think they should purchase right from the very newly manufactured RMP lots and half expiry completed lots and near to expiry lots. So when you take all these ages into consideration, definitely it would be helpful to cover all the variations.
Right. Okay, looking at the time, I think thank you very much for those insights. I think it really provided then some pretty specific recommendations also, and answered some questions around the applicability of some of those ideas in the paper. So thanks for that. And we might also, if there's any further questions, feel free to post them during the next presentations. But with that, I would then hand over to our next speaker, who's Dr. Parvinder Punia, on the regulatory strategies for biosimilars, the past and the future.
Okay, so yes, I have a background in regulatory affairs and leading a team of CMC clinical and non-clinical experts. We have quite a lot of experience in biosimilars. I won't go over the origin of what is a biosimilar. I think that was covered very well in the previous presentation, and I will try to make back some time for you if you want to have a longer discussion at the end. The origin of the guidelines in EU at least is since 2004 with the first publication, and even I remember it during my career, and it was quite revolutionary to see this that the EMA had come together nearly over 20 years ago and created the first biosimilars guideline. The first biosimilar that was actually approved was 2 years later in 2006. So it's, we're looking at a 20-year history and experience of the EU of approvals of biosimilars, which is significant. Around the world, the EU is not the only regulatory body with the matured guidelines. All stringent regulatory jurisdictions have developed their own guidelines or are closely observing those of others. The observing countries are following the guidelines from agencies such as the EU, FDA, and or the WHO. So the importance of the EU guideline and its regulatory framework, I believe, is quite important for worldwide developments. Therefore, the EU will probably be one of the facilitators, I hope, of the harmonization of the global guidelines. And as was mentioned in the previous talk, and it will be mentioned again in this one, the new reflection paper, I believe it's a movement in the EU, but it will be quite closely observed by other jurisdictions. I think everybody knows this image. It's the basic snapshot of the developments and the data's requirements for the original reference biological product against the biosimilar. And as you can see, and as was mentioned in the previous presentation, and I guess it will be mentioned in the next one, the structure is the same, but the weight of evidence is quite different. The original biological has the evidence heavily reliant upon the non-clinical and the clinical studies, obviously, because they're proving in their product in an indication. But the biosimilar has a mixed weight of evidence for quality and comparability at the analytical part, as well as the clinical studies and a peppering of the non-clinical studies. This is currently how it stands now. As you can see, there is still the clinical burden on the biosimilars, which is very much quite different to small molecule generics. So just to set the tone, and this is just a snapshot, and I could have done 5 slides of this, but I only managed to get 2022 to 2025 into a single slide. This is really a one-liner analysis of the type of data that was included in significant biosimilars approvals in the last 3 years. And as you can see, all of them have contained clinical studies. They do have a mixture. There is a lot of analytical characterization, but confirmatory efficacy studies, essentially phase 3 studies, are contained in the dossiers in MAs that have been approved. So over the years, there was a lot of experience gathered from the agencies. My team here, they're all ex-assessors. So this is gathered anecdotally from inside and outside the agencies, and additionally from a very interesting paper that was published in 2023. And the authors of this paper were from the regulatory agencies that have been highly involved in the assessment of biosimilars, such as Paul Ehrlich Institute, the Austrian agency, and a number of other agencies. And in this paper, and this was the paper that was cited actually during the current revision of the reflection paper. So it's a very insightful study. But there are other studies as well. In this paper, there were 33 monoclonal antibodies and 3 fusion proteins that were submitted to the EMA between July 2012 and November 2022. I think that's quite a good amount of time to see how the evolution of the assessments have gone at the EMA. I encourage everybody working in biosimilars to read this paper. What they did was they analyzed the role of quality and CMC derived data and what was the weight of that data on the conclusion of biosimilarity in the marketing authorizations. And this was analyzed against the clinical evidence of those applications. The special emphasis has been placed on 11 biosimilar candidates that are listed here on this slide. And this evaluation was all taking taken place retrospectively, so that all finalized reports and approved marketing authorizations. There were, however, 2 applications withdrawn, which I will discuss later. I think it's important to note that when an application was withdrawn, usually in my experience, it's a prevention of a rejection. So they were not withdrawn for any other reason except that they were not considered complete enough to meet the biosimilarity requirements of the time. The paper has categorized very broadly, so it's not so deeply categorized, the approval of the products as acceptable, non-acceptable, or that there were some uncertainties in the data, but that didn't block the approval. I think what's really important about this paper, and which I encourage everybody to read, is that it examined the assessment of the day 120 reports of the procedures. This is not public information. This is information contained within the agencies. But it serves as a deep information of these packages of the data evolution for the approval of biosimilars. As a disclaimer, I would say that if you're looking at a day 120 assessment report and then you're reading the EPAR, there's a lot of changes that go on between those 2 time points in a procedure. But what the day 120 assessment reports do serve as is the sort of first data look at the original development program of any of these biosimilars. Of course, anybody who's worked in biosimilars knows that what goes into the day 120 report and the dossier that came before it can be quite different to what happens during the procedure and the data that the procedure ended with, depending on the major objections that are received during the procedure. What happens at that time point during the procedure between day 120 and the end of the procedure is really very much at the discretion of the agency and the applicant. So that's not captured in this paper, but you can probably extrapolate quite a lot from the EPARs. So they summarized it in this way. For the 36 monoclonal antibodies and fusion protein biosimilar candidates, the quality CMC, i.e. the general aspects and analytical comparability exercise, clinical PK/PD and clinical efficacy, safety and immunogenicity aspects were analysed on the information provided in the EPAR. Results are shown here to the 5 possible scenarios that have occurred. I didn't annotate the last scenario because it's a complete rejection and there were not any in that case. For more than 80% of the biosimilar candidates, the quality CMC part of the dossier, the clinical PK/PD and the efficacy, safety and immunogenicity were all unambiguously supported by similarity. So these were very big programs of development. For 2 of the biosimilar candidates, differences in some QAs and functional assays were observed by the agency, but these differences did not cross over to PK/PD and the efficacy, safety, immunogenicity conclusions. One candidate did have higher immunogenicity, but they later deemed it irrelevant. And all of these biosimilars are listed as approved MAs under scenario 1, so they had very comprehensive programs. The second set of monoclonal antibodies that were classified in this paper were 2 cases that had rather unsatisfactory quality in CMC, but they had overall acceptability in clinical trials. In these 2 cases, concerns had been raised regarding the biosimilarity as well as the comparability of the clinical batches and the commercial batches of the biosimilar. So this maybe relates to the last question that we just answered. The clinical PK and efficacy trials met their endpoints. However, there were still some uncertainties for the clinical efficacy regarding the secondary and the subgroup analysis for the biosimilar candidates. Both applications were withdrawn by the companies owing to the major remaining concerns. Scenario 3 was defined as those products that had an acceptable quality CMC, but they had differences in PK/PD, or they had remaining questions regarding test materials in the PK study, while all other clinical data was demonstrated. 2 of the biosimilars were analysed in this, which was very interesting, had failed PK studies. In both instances, it was argued that the differences observed in glycan structure were known to affect the PK, but they were too small to explain the initially observed PK differences. They conducted second PK trials with an improved design, and they were able to show demonstration of similar PK profiles with the reference product. For the third biosimilar in this group, PK results were not accepted because the test product was not deemed representative of the commercial product. In this group, approval was given to these biosimilars. In the fourth scenario, these were the cases which had a very acceptable quality and CMC package. They'd successfully completed the PK, but there were issues in the efficacy, safety, and the immunogenicity data. For both of these biosimilars, the primary efficacy endpoint was not formally met, as the upper limit of the confidence interval was not contained within the predefined margins. So for both of these, an MA was actually granted because of the convincing quality CMC and PK data, and that the agency deemed that it didn't affect the safety, efficacy, and the immunogenicity profile of the biosimilar. The last scenario, which was a hypothetical one, was completely unconvincing data in quality CMC or completely failed clinical trials, and it wasn't observed in any of the biosimilars that were studied in this paper. I think overall this paints a picture that the approval requirements, as you can see, can differ depending on the program. And I think it relates back to what was said earlier, that if the mode of action is well defined for the reference product, there can be some flexibility with regards to what the development could look like. Basically, possibly taking the weight off phase 3 clinical studies, as is now being discussed. Just for people who have never done a biosimilars procedure, we've done many. These are the kinds, and the paper also reiterates these are the kinds of major objections that an agency gives for biosimilars applications. These are most common, I would say, and they're also listed in the paper. They can be anything that would be regarding formal aspects such as GMP, etc., but most of these usually get solved during the application process with a conversation with the EMA, delays in restarting of the clock. You can get the GMP's inspections done. In other areas, the biosimilarity exercise are sometimes deficient in aspects such as insufficient batches, as was also mentioned earlier. The ages of batches is very important, or incomplete testing panels, amongst other concerns. For general quality, the process definition or control, or the consistency of the processes can be deficient, or they had generally missing data. With regards to the clinical differences, often where the PK differences are not well explained in the clinical context and can be very different to explanations that we see for generic developments. So the extrapolation to the indications can be challenging, and there are numerous cases of the reference product having numerous indications, and if you don't have a good PK bond with the reference product, the extrapolation of the indications is a challenge. Then there are other aspects, formal aspects in clinical, such as GCP, which is quite common these days. So please be careful with your GCP requirements for the clinical studies. And then, of course, the big one that's always discussed and I see this question in any shape or form in biosimilars, which is the immunogenicity question. The applicants are often failing to provide sufficient long-term extrapolation for the immunogenicity profile. And then the really, really serious concerns are which there's just been complete failure to meet primary endpoints in a phase 3 clinical study in comparison to the reference product. But as you can see, and these look quite major when you look at them, most of the approvals from the previous slide that I showed are approved. So these are quite serious concerns in a biosimilar, and these are major objections that appear at day 120 of a procedure. Yet, hence, they should be challenging to respond to, but actually they are all able to be managed and responded to. to the point that most biosimilars do actually get approved, even with these kinds of questions in them. So when you see questions like this, I think the first thing you must not do is panic. What the agency wants to see is a practical link to these deficiencies on basically the gold standard question that the CHMP will ask, which is how does this impact safety, efficacy and immunogenicity of the biosimilar? That, having said that, that doesn't mean that if you haven't done enough biosimilarity or you have not done enough reference to test product batch analytical similarity, of course, they would lead to failures in biosimilarity. These are the two withdrawn candidates, and as you can see, I think you don't need to read all the details, but as you can see, the slide can show that they were failing. It's a pitch patch of failed parts of a development that would never have led to a procedure to be approved for this kind of biosimilar. The QTPPs were not adequately established, and there was insufficient data from test and reference batches. Overall, the comprehensiveness of these data packages were severely deficient. I think that it's often the case that programs that have been approved in other less regulated regions often are submitted as is, maybe with some supplementary information against the European reference product to a centralized procedure in the EU. And these kinds of developments are usually not satisfactory for the requirements of the EU. So that's a place to step carefully, even if a product has been accepted in a region that it's not recognized at the ICH standard, the EU will not approve it. So this was the first rethinking of biosimilar approvals that had happened. The study of this paper showed that the regulatory decision making is really based on the totality of the evidence. This is a nice phrase that's been used in the last few years to make the regulators rethink about how they look at packages of complex molecules, not only biosimilars, but there are other complex molecules with similar requirements to biosimilars. And that the main focus on the pharmaceutical quality and general quality and the PK similarity aspects could be quite important. And therefore that the clinical efficacy study may not be requested or needed. In the study, 29 out of the 36 cases had really good quality CMC packages, and they were matched with successful clinical trials leading to the marketing authorization. But interestingly, the good quality CMC packages could also be paired with failed efficacy or PK studies, which were evaluated due to be reasons not related to biosimilarity, the biosimilar candidate. Thus, the agency gave the marketing authorization. On the opposite side of that, unconvincing quality CMC data with the successful clinical trial also didn't allow for the marketing authorization, because they primarily due to the lack of the demonstration of sufficient pharmaceutical quality and functional similarity with the reference product. There was only one case where the clinical data analysis led to a major objection, and there was a divergent position, and it's been published. And I haven't linked it here. Sorry, it's a mistake in my slide. I can link the report where you'll see the divergent position. But they received a marketing authorization. As a summary, the authors of this paper found that the reports show that the quality CMC played an important role and was predictive actually for the approval. These findings led to a conclusion now that a reduction of the clinical development could be applicable. These products have since gained more data from the market, and the earlier predictions have been supported. As a consequence, a revision of the regulatory guideline has taken place. What would be interesting really is to be able to, maybe in 4 years' time, since these approvals have come along, see what data has occurred. It would be nice to have another publication of the feedback of the post-marketing experience from some of these applications from this paper. But there is a concern on the other side, so I'll play the devil as well on one side. That there is concerns regarding allowing biosimilars to be approved with no or minimal clinical efficacy evidence. The stakeholders such as patients, physicians, they do still have their concern about switchability between the reference product and the biosimilar. But this paper has been partially successful, from my understanding, to alleviate those concerns, and that the totality of evidence approach, which focuses on the quality in the CMC and the PK similarity, is applicable for biosimilars. Again, I repeat, in the case where the mode of action is quite well defined of the reference product. And therefore, and it goes back to Stephen's introduction, the routine and costly clinical efficacy trials are really time consuming, and they may be a huge barrier to encouraging biosimilar developments where they are very much needed, considering the patent losses that are coming up with the reference products. Subsequently, I hope you've all read it. The reflection paper on the tailored clinical approach in the biosimilar development has been set up for public consultation. You have until the 30th of September. I do encourage anybody involved in biosimilar developments to contribute to this public consultation. It's interesting because the reflection paper is on a tailored clinical approach, but 14 pages of this 17-page paper is actually on CMC and quality methodology, and the basis is not clinical at all. So this is a big pivot of the European agencies, and maybe this will become a global gold standard in the future. So I think that this is a really important time for companies engaging with the EMA to have a say in what would be considered a good program for biosimilarity. The paper also had other findings that suggested that full clinical trials are also not necessary or even not sensitive enough to detect the dissimilarities. I mean, I personally feel that when you do a clinical study, you're looking at one snapshot in time in testing, but when you are looking from the quality perspective and the ongoing quality programs that biosimilars have to take in, considering the variability that can happen, it doesn't make any sense to base all the biosimilarity on just a clinical trial that took place at one point in time. Additionally, and I think that the agencies have learned a hard lesson here, I can tell you that because my team was from the agencies, the laboratory-based characterization tools are very advanced. They are now also being recognized by the regulators. I have myself been in scientific advice for biosimilars many times, and often the assessors have had their curiosity piqued when we have been talking about some of the new state-of-the-art methodologies that have a better proof of biosimilarity than clinical would have. It is again notable that the mode of action of most reference products is known, and that PK still remains a good option for those types of products. It is quite important to recall that so many indications are approved for some of the reference products, so it shouldn't remain the burden of proof of the biosimilar company to prove the indications. And additionally, we mustn't forget that there are many product-specific guidelines still already available, that can aid the developments. And also that the PD markers and an effective quality comparability can be managed for most of these biosimilar candidates. So the agency has will encourage the use of the tailored clinical approach. They are open to offering advice in the tailored scientific advice program, but there is of course a question of strategy. When do you go to the agency? I think the previous speaker probably has much more experience, but one of the challenges that I've seen is during the scaling. Companies spend a lot of money when you scale up. And do you go at the point after scaling with the sort of a pre-assessment of your program biosimilarity, even from the quality perspective, because that is also costly, or at some point before the scaling process to to the commercial scale. There's still a lot of ambiguity in the paper, and I think it's really experience-based, and it's based on the capabilities of the company's development about when they go for approach to the EMA for scientific advice. They do have the tailored scientific advice for biosimilars, but again, it doesn't give clear advice to the sponsors, and I think that's because the agency itself doesn't have the answers about when to approach them and the timing that you would use, at what point during the development you should go for the tailored scientific approach. One must remember that the SAWP process, so for somebody who hasn't done that, that's a scientific advice working party process. It's very long and costly. For a biosimilar, for all development aspects, a single SAWP opportunity costs just under 90,000 euros. The process can take anything up to 4 months. So how does that fit into a biosimilar development? It's a very long process. People are not sitting twiddling their thumbs waiting for an answer from the agency. They want to get forward with scaling. They want to start creating the batches for the studies. So we spend a lot of time working on the data at some point in time when we think it's an appropriate time to go for scientific advice. And of course, the part would be about the scaling. When do you go to talk to the agency, and do you go multiple times at that cost and that time? It's quite a lot in terms of cost and time to keep going back to the European Medicines Agency. Previously, we would go, for example, for national advice. So this is something historical that my team have done, that we wouldn't go straight to the EMA. We would go for national advice. You'd go to somewhere like Germany, which is anyway one of the main rapporteurs for biosimilars in the EU. For national advice, it's much more flexible. You get a face-to-face meeting. It's cheaper, and you can turn around this type of advice in 6 to 8 weeks. And the development can keep going. So I'm a little bit hesitant about this one-size-fits-all approach in a regulatory strategy for the development of a biosimilar using the tailored approach. And often at the CMC level, there are challenges, and one must think, when do I go to the agency to see if this challenge is insurmountable from the regulatory perspective? Because discarding the entire program is not an option. And also, what is the differences that I'm seeing at the analytical level? What is their clinical relevance? And this is something that needs to be discussed with the agency before you continue on with the biosimilarity, buying more reference product lots, generating more data of the biosimilar product. It's very costly. And of course, all of these questions are always open to the interpretability of 28 members of the EU about what would be the immunogenicity extrapolation. So that should be considered. And last but not least, often there are post-marketing commitments. So ongoing safety collection should be part of your overall plan when you talk to the agencies. The EMA has I won't spend too long on this, you can read this on the EMA website. The EMA has completed the pilot program. It ran for quite a long time, but only 4 applicants submitted altogether 6 advices. So you can see that there were multiple advices from one candidate. One applicant was an SME, so I guess that meant that they didn't pay so much, so they probably went multiple times. They don't pay 90,000 euros each time. There were 8 more applicants that expressed interest, but it was not clear to me why they didn't follow up in the tailored advice, and they were all for monoclonal antibodies. The key topics, and if you go back to the major objections, they match. They're waiving preclinical studies, so I guess talking about the analytical comparability to waive preclinical studies, method development, and the quality differences that are impacting the PK/PD and the safety and efficacy. So the extrapolation of any quality differences, how they will be in the patient. And the big one, of course, is reducing the clinical studies. And many companies have been successful to reduce the clinical studies. They finalized this pilot in 2020. At the same time, 71 standard advice requests were taken. So imagine 6 in the tailored and 71 standard advice for biosimilars. And I think it's really important to remember this is only with the EMA. We don't know from these findings if the same programs went through national advice before they came to the EMA. And that was a very common pathway a few years ago. So the EMA advice would usually be the later advice you would do in order to sort of seal the deal or find out some differences from them. But most programs would have gone from 1 or 2 national advices before that. They did feedback that they liked the in-depth dialogue with the regulators, but the SAWP as a standard doesn't give a meeting, so that would be interesting to see how that will go in the future. There have been 2 approvals since the pilot has, from the pilot advices. These are listed here. Oh, I think I lost this. Did I lose a slide? Wait. This is a busy slide. I will leave it here, or I can share it with somebody. We use it as a standard for big discussions on immunogenicity. It's the two other big deals that we see during biosimilar assessments is long-term immunogenicity, and the other one, which I'll discuss later, is the extrapolation of the indications. These are usually the big topics during scientific advice and assessments, amongst others. Right now, the clinical study is considered the method of detection with the continued follow-up. The goal of the study, of course, is to detect and compare immunogenicity under the real use conditions against the reference product, and it's supposed to be directly compared to the reference product, and the clinical study does that. The regulatory agencies have a complete expectation, this FDA, EMA, everyone, that there would be no clinically meaningful differences. And if the differences are observed, they do need to be either investigated or explained. So often in the procedures that I've been involved in, immunogenicity question will last until the end of the procedure. And this means that even if it's considered a minor point, there's still a lot of other parts of the dossier that you have to satisfy for that question to disappear. It's often not a standalone question on its own. There's a lot of other parts of the dossier that answer this question. And then even after the approval, you can suggest, of course, but that depends on how much risk the agency sees in the immunogenicity, that pharmacovigilance and real-world data can continue to monitor the immunogenicity, because it's true that most rare immunoresponses will not occur during the trials. So this is another reason why efficacy studies are not fully answering all the questions of a biosimilar candidate. But there seems to be now a bit more of an understanding that the clinical trial could be minimized, and specifically could be for immunogenicity if analytical similarity is high. PK/PD studies could be considered in healthy volunteers, for example. And also, I think that this is the big one, that the reference product has low immunogenicity risk. But of course, you will always have to provide a strong scientific justification if you are not able to prove unequivocally risk of immunogenicity. And in other cases, I think particularly in oncology, but it happens in all, there are big problems with regards to extrapolation of indications. You cannot conduct a clinical study across all indications. It's impossible. But the extrapolation is possible if it's supported by totality of evidence. Again, this is a question that usually gets answered after everything else has been satisfied in the dossier, and that's could be PK, it could be clinical, it could be quality. However, I think that this has become a little bit more established now, and has been quite well demonstrated in many approvals. And I personally, and I don't think that there has been a need to repeat any studies for other indications in the approved biosimilars. There is evidence to support biosimilars developments without clinical comparative efficacy or biomarker PD studies. The regulatory sciences for biosimilar has evolved, and EMA is leading this initiative to improve and streamline the development without the clinical comparative efficacy study. Now, there are a number of other papers. We can give you the list. If you go to the paper that I've cited earlier in this presentation, in the reference, you'll see other papers that have really taken a deep dive into the data requirements that have come through approved marketing authorizations. And there are other agencies now following suit, such as the MHRA, with its own guidelines. So I think that EMA will lead, I hope, in improving the program for biosimilars. So overall, I mean, this is the weight of evidence maybe will be streamlined with the emphasis being promoted for physiochemical analysis, and hopefully less onerous requirements on the clinical study. But we will see after the stakeholders have done their consultations. So the streamlining, I think it's necessary. There is regulatory science and existing data. This hasn't been made up. I think there's been a really deep analysis on many dossiers by the regulators themselves. So the regulatory science really supports this change. There are still many biologicals that do not have biosimilar competition. So I think that's a big thing with regards to the payers. The streamlining is really needed for the expansion of competition and to save money at the healthcare level, and to increase the access to biologicals, because right now it's decreasing, it's prohibitive financially in many countries. So this just to say thank you to everybody, and the evolution of the regulatory framework towards the tailored approach is looking like it will shift away from clinical studies, efficacy studies, and that there will still be a high standard for biosimilarity. And I think that the main thing is that the doctors and the patients accept this standard, because it's difficult to digest that you would switch from a biological reference to a biosimilar. But with all of the weight of evidence now in the scientific domain, that the risk is quite minimal if you meet all the parameters. I think that there should be no reason not to support the change in the regulations. Thank you. I will stop there.
Great, thank you so much. I think a lot of great additional insights. Also, really the big important questions about how to phase now, seeking the scientific advice. So I think lots of interesting input there. Thank you very much. For the interest of time, I do want to be mindful of time, perhaps, and I didn't really see now any right now additional questions. So therefore, I would suggest if Jiří can continue with this presentation, which is around patient safety and biosimilarity trials, and then we can hopefully still have some time at the end for additional questions.
Hello, everybody again. My name is Juchelka. I'm the principal investigator at Quinta-Analytica, working in Ostrava and Prague clinical units in Czech Republic. And since we have really mixed audience today, a lot of people from pharma industry, experts from clinical medicine, academia, etc., clinical research, and you have already heard two excellent presentations about the analytical similarities and the regulatory basically approaches with regards to biosimilar development. I would like to talk more about the biosimilar safety monitoring, immunogenicity. You have already heard a lot about immunogenicity, why it is so important, why it matters so much. And we will touch on some safety monitoring concerns, and also, because I'm a clinician and an investigator, I will speak about what are the expectations from regulators and with regards to pharmacovigilance from us as investigators and clinicians. where it really starts from, the biosimilars differences, they really start at the manufacturing process. And you have heard a lot about that from Thakara. The three molecules which you can see on the pictures are basically identical with regards to the core amino acid chain. But as you can see, there are slight differences with regards to these little blue chains, which are sugar chains attached to the core chain. And these can be either three different biosimilars, or these can be differences batch to batch. And the yellow highlighted spots actually shows the little nuances, which we call microheterogeneity, and those can have impact on structural and functional variability, as well as on immunogenic potential. And to be even more complicated, these nuances and these impacts may differ across different indications. They may even differ from patient to patient. They differ in different routes of administration and so on. These two pictures show an abbreviated development of a reference biological medicine and the biosimilar on the right-hand side. So for the original biologic, you can see it starts from the analytical and preclinical parts, but really vast testing, vast clinical research is done in the clinical phase, from clinical studies, phase 1 through phase 3. Whereas for the biosimilar development, big emphasis is put on the analytical and preclinical parts. And the clinical PK/PD and confirmatory clinical safety, efficacy, and immunogenicity studies are much more abbreviated and, let's say, different in terms of extensity when it compares to the original biologics. Biosimilarity is assessed as a global, but safety, efficacy, and immunogenicity is always part of really deep scrutiny by the regulators, and it's very important to demonstrate that there are no differences or significant differences between the original biologic and the biosimilar with regards to safety, efficacy, and immunogenicity. You have seen this really interesting picture presented by Parvinder earlier today. We must understand that biosimilars undergo an abbreviated clinical development pathway compared to the innovator drugs. And these clinical trials, which are performed for safety, potentially efficacy, PK, PD, and immunogenicity, may be done just in one indication quite often, because there is this extrapolation of indication effect. They involve fewer patients than large global clinical programs for the original biologics. And pharmacovigilance of a biosimilar must really address all these potential deficits in order to provide sufficient evidence and sufficient safety, immunogenicity, and potentially efficacy data in order to prove biosimilarity with the reference product. What is, of course, very similar for both original biologic, and the biosimilar medicine is the further risk management, the risk management plans, and the extensive long-term ongoing pharmacovigilance program, which starts really before the marketing authorization, but it spends long time across the entire life cycle of the molecule in order to bring new data and show potential safety signals or immunogenicity signals which were not detected during the potentially short clinical development program of the biosimilar. There are some specific concerns which we also consider when talking about impacts on safety and efficacy. We have already spoken about microheterogeneity, structural, functional variability. However, when the drug is going to the market, when it's already approved and it's being manufactured, there still can be changes which are sometimes unintentional, unwanted, unexpected during the manufacturing process, which can cause product drift or product evolution or even product divergence. These are basically excursions in the structural or functional specifics of the compound. If they are still within the acceptability limits, it's quite okay. But if they extend beyond the acceptability limits, they need to be tested for potential safety, efficacy impacts, or immunogenicity. And if they really are significantly different from the original comparator product, then we call it product divergence, and the two molecules are no longer interchangeable, no longer considered as biosimilar. Changes can happen when various batches of product are manufactured in different manufacturing plants, for example. And what we as clinicians see very often when the biosimilars are already on the market is also switching or interchangeability. This comes from the fundamental nature of biosimilars, because biosimilars should be interchangeable with their original biologic reference product. And it really happens very often in clinical practice, because we see shortages in market supply or reimbursement issues, which may require the clinicians to change one biologic product with a biosimilar, or a biosimilar to another biosimilar. And all these concerns, all these areas actually may have an impact on immunogenicity of the product, which then subsequently impacts potentially safety and efficacy. Microheterogeneity of the products can impact the biological activity of the product. It can negatively impact pharmacokinetics. It can increase clearance of the product or decrease stability. And it may also reduce the ability to bind to the target receptors, and therefore potentially it can affect the efficacy of the product. Structural variability, such as post-translational modifications, may lead to occurrence of new epitopes on the surface of the molecule, which can trigger antibody formation and potentially antibody-related reactions. The batch-to-batch variability, sugar chain composition changes we have already spoken about, as well as about the product drift and evolution. So all these changes may impact safety and efficacy of the product. Generally speaking, majority of adverse reactions and adverse effects of biosimilar drugs or biologics are derived or dependent on their pharmacological background, basically, and mode of action. Only a small percentage of adverse effects and adverse reactions are immune-related. However, those adverse reactions which are associated with the pharmacology of the drug, those can be quite well anticipated, they are quite well described, and they can be quite well extrapolated from the biological original product. Whereas immunogenicity can be quite unexpected, and even small nuances and small differences between the original biologic and the biosimilar may lead to occurrence of anti-drug antibodies and immune associated reactions. Why is that? Biologics and biosimilars are large, complex proteins. The larger, the more they can induce immune responses and form antibodies such as anti-drug antibodies. When you look at these pictures here, you can see a molecule of aspirin, which has a molecular weight of 180 Daltons. Then the three different biologics, insulin, growth hormone, and monoclonal antibodies, are much, much larger molecules. Monoclonal antibodies can have molecular weight about 150,000 Daltons, so it's really huge, and the potential for immunogenicity is really much, much bigger when compared to the others. Anti-drug antibodies are important because they can block target binding, they can increase clearance of the drug from the organism, they can induce drug aggregation, and so on and so on. There are various aspects or factors which contribute to immunogenicity of the product. Some of them are drug-related. It's the product origin itself, its structure, modification of the molecule during the manufacturing process, its mechanism of action, potential impurities in the manufacturing process, as well as formulation, packaging, etc. And then there are a lot of aspects or factors which are patient-related or disease-related. It's the disease itself. Some diseases may have higher immunogenicity potential. Patient to patient are different, so patient population also play a role. Prior sensitization, whether the patient has already used any similar biosimilar or biological products or has any history of allergies or allergic reactions. Patient's genetic status, age, status of immunity or immune tolerance. What is also very important to address and to test is the impact of route, dose, and frequency of administration. For example, we know that intravenous administration of biologics is less or typically less immunogenic than subcutaneous, intradermal, or intramuscular administration. Or if you administer a biological product multiple times, it increases the potential for a delayed immunogenicity and delayed immune reaction. Also, the window, the drug-free window between one and the other administration may have immunogenicity impact. Duration of treatment, concomitant medication, so a lot of factors which actually contribute to this complexity. If there are immunogenicity concerns, they may have some safety consequences. Luckily, this is typically very rare. So even European agency and regulators are not that concerned with regards to occurrence or incidence of these immune-mediated reactions because they are really, really very rare. However, as you have heard from Parvinder, immunogenicity is always very much scrutinized, and there were many questions with regards to immunogenicity in the deficiencies of the dossiers. Actually, many more questions were regarding immunogenicity than regarding the general safety of the compounds. Acute adverse event reactions, short-term reactions, can be very mild to life-threatening. It can be something like erythema or injection site reactions, local swelling. It can be rash, it can be flushing, but it can be also severe and potentially life-threatening reactions, anaphylactic or anaphylactoid, which may potentially lead to life-threatening conditions such as hemodynamic shock. In long-term perspective, there can be immune-mediated disorders which may start or which may be detected months after the administration of the biologic drug. And this is when anti-drug antibodies start neutralizing endogenous proteins which are or look similar to the drug. This happened, for example, with biosimilar erythropoietin, where anti-drug antibodies actually started neutralizing the endogenous erythropoietin, which caused severe hematologic disease called pure red cell aplasia in patients. There can be cross-reactivity. Anti-drug antibodies may start reacting with some antigens in the organism, which can cause autoimmune reactions. Some of them, again, potentially very severe and life-threatening, such as rheumatoid disease, lupus-like syndrome or similar. And they can also decrease immune surveillance and increase or put the patients in an increased risk of infections and malignancies. If anti-drug antibodies bind to the drug itself, it may block interaction with the target, and it may have negative effect on the therapeutic effect of the drug. I have already mentioned that drug clearance may be enhanced. There can be lower circulating drug levels. This is something which is, for example, known for TNF-alpha antagonists. Infliximab, biosimilar infliximab, was demonstrated that the patients using this drug have actually very high titers of circulating anti-drug antibodies and quite low percentage of intact infliximab after administration, which is efficient. There can be cross-reactivity with other products, and the anti-drug antibodies can also affect some specific reactions or modes of actions, such as oncology, monoclonal antibodies, which cause antibody-dependent cellular cytotoxicity. Regulators typically look at the immunogenicity in the totality of evidence approach. They require head-to-head comparison with the reference product through ADA testing using validated assays. However, they also consider each of the molecules with regards to their risk because, and this was already mentioned by Parvinder as well, there can be some molecules, especially the low size or smaller size molecules, such as heparins, such as some insulins, such as granulocyte colony-stimulating factors, where the risk of immunogenicity is much lower than, for example, with monoclonal antibodies or fusion proteins. And there can be even some waivers granted for immunogenicity testing in those types of molecules. How is the testing performed? This is a very abbreviated scheme. It's normally much more complex. However, it's a multi-step process performed in tiers. In the first tier, we do highly sensitive tests to detect any potentially positive binding anti-drug antibodies. Those which are positive go to tier 2 testing, which is done through highly specific tests to confirm whether they are really positive or negative. And those which are really positive go to the tier 3 testing, which is a titration assessment. This is done again through highly sensitive tests. And also, those positive anti-drug antibodies undergo specific testing with specific assays to detect whether they are neutralizing antibodies or not. Neutralizing antibodies are can be clinically very relevant because they bind to the drug, they interfere with the target, and they potentially reduce efficacy of the drug. And all this antibody or immunogenicity testing and examination is then presented through various tables and listing to the regulators. We also need to consider what type or what design of clinical study is the most appropriate for immunogenicity testing. Sometimes crossover studies are not the most suitable ones for immunogenicity testing because generation of anti-drug antibodies can be a very lengthy process. They can really appear months or even a year after the study drug administration. The half-life of anti-drug antibodies is very long, and typically the washout between periods in the crossover studies is not sufficient to distinguish whether the antibodies which can occur months later were actually generated by the tested product or the reference product. Therefore, for many of those immunogenicity studies, we prefer the parallel design. However, again, some small molecules or low immunogenicity risk molecules may be tested in crossover studies as well. What is very important in terms of pharmacovigilance of biosimilars in general is long-term post-marketing monitoring. There are many specialized needs and requirements how the gaps or deficiencies in data from the clinical development need to be compensated throughout the post-marketing pharmacovigilance and safety monitoring. It all starts with tailored risk management plans, which must be very robust and must address all potential risks, concerns, and known immunogenicity or safety risks extrapolated from the biologic original product. What is very important is traceability and identification of adverse effects and immune-mediated reactions. Because each batch of biosimilar products, each biosimilar itself, each biologic product itself may cause slightly different immune reactions or adverse events. And when we collect this information, either actively through medical records, drug and disease registries, or patient-reported surveys, or through spontaneous passive surveillance systems from institutions or from clinician reports, or through literature searches, media monitoring, etc., we always need to trace down the adverse events and the immune reactions to the exact individual batch of a biosimilar product. We cannot put them all into one basket because we really need to know and understand why exactly this product and this batch was, let's say, more immunogenic than the other. Risk management plans must extrapolate all risks from the reference product, as well as must encompass all risks and specific concerns, immunogenicity and safety concerns, which are already known or anticipated for the biosimilar itself. Sometimes there must be specific post-authorization safety studies performed in specific patients with specific diagnoses, with specific indications, and so on. What is, of course, mandatory is the periodic reporting of safety to the regulators, to the authorities, and the periodic evaluation of the benefit risk report, which may change during the time, especially if we find out some previously unknown safety or immunogenicity concerns. We must adopt or plan risk minimization measures. Therefore, patient educational materials or professional educational materials are being developed. Sometimes there are special patient registries formed in order to collect data about immunogenicity throughout the long-term use of the product, special cohort monitoring. And whenever there is any new information which may impact the benefit safety risk, this must be immediately communicated to healthcare professionals and to patients themselves. And you probably know this black triangle symbol, which is a symbol in European labeling for all drugs which are still in safety monitoring and which still undergo additional safety data collection. What is expected from us, from investigators, from clinicians with regards to safety monitoring? We must first understand how heterogeneous is our study population, whether we have treatment-naive patients or whether we have patients which were already pre-treated sometimes in the past. What is surprising is that pre-existing antibodies, which sometimes can be detected even in treatment-naive subjects, may exist in the population. Sometimes we don't have any idea how they appeared, where they came from. But the testing for the pre-existence of antibodies in our study population is very important. Parvinder already spoke about sufficiently long observation periods because it can really happen that the safety signals will start appearing months after the time when we treated this patient population. Adverse event tracking and traceability I have already spoken about. What sometimes happens is the adverse event underreporting, especially when it comes to rare or late adverse events or delayed or non-acute manifestations. Clinicians typically focus on acute reactions or short-term reactions. However, the longer is the study and the longer is the gap between the study drug administration, the last study drug administration and our monitoring checkpoint, they tend to neglect some mild or moderate reactions or adverse events which the patients may report to them. And those can be actually safety signals or immunogenicity signals which were not known before and started occurring only at that time point. If there are any new risk updates, investigators must be timely informed about those, and this not only concerns the biosimilar itself, but also any safety signals or new safety information which relate to the original biologic, and they must communicate these new risks, contraindications, and precautions to their patients. And even though I have mentioned that life-threatening reactions are very rare with regards to statistical point of view, we still need to be prepared. We cannot be surprised when something unexpected happens. Always we need to have emergency measures ready, emergency equipment ready, and must be ready to provide life support to our patients or study subjects if something unexpected happens, because even though the risk profile of the original biologic can be satisfactory, we can never 100% extrapolate, and there may be something which would be unexpected with our biosimilar, which may occur at this point. I will finish my presentation with this statement or citation that immunogenicity is like a bouncer at a club. So if there is a molecule which shows up looking suspicious or too different, the bouncer would kick it out. But if it looks familiar or too friendly, it gets a free pass and might even get a VIP welcome. And this is what we need with our biosimilars and with our patients. We want them to get a free pass and a VIP welcome when we administer biosimilars to our patients in order to provide our patients with the deserved benefits of the biosimilar products. Thank you very much for your attention.
Great. Thank you very much, Jiří. Just, right on time at 15:59. I think right now there was no additional question. What I would like to encourage anyone is to get in contact with us with any kind of additional questions. We at Conscio Group, are in the great position to, provide both analytical testing, but also the clinical studies on healthy volunteers. So we are excited about staying in close contact with you and really considering this journey with you. So if there is any further questions, please feel free to contact us or, and follow us also on our LinkedIn accounts, contact us personally. We'd be more than happy. And with that, I want to thank all of our speakers again today for the interesting discussion. Stay tuned. I think what we will plan is also to make this a regular interaction, because we feel there's a constant, steady flow of news for us to share with everybody. So we'll probably have another edition of our webinar series later in the year. So again, thank you very much all for attending, and stay in touch. Have a great rest of your day.